Can you hear me? If I'm audible, can I see a thumbs up, please? Good evening, Dr. P. Am I audible, Dr. P? Yeah, thank you. So, if you're joining my session for the first time, this is Dr. Shanmugapriya. I'm Professor and Head of the Department of Biochemistry at Government Thuthukudi Medical College. Good evening, Skull. Good evening, Dr. P. Good evening, Nidhi Jan. And I'm an Unacademy educator for biochemistry. And today's session is a biochem recall. I will be recalling all questions. Based on your recall, I will be discussing biochemistry questions which were asked in the latest INI-CET May 2022. Hi, Vanilla. And I do agree that most of these questions were unexpected, and there were hardly any questions on metabolism or chemistry. It was completely a molecular biology aspect of biochemistry, and most of these questions probably were the first time, right? It was the first time that they had asked such topics. So this does not mean that you should get disheartened or you should feel depressed and stop preparing for NEET PG 2022. All that I want you to know is both are two different institutes, right? It's both are two different organizations. So the way INI-CET has been, the question paper has been set is not going to reflect anything on how or what you can expect for NEET PG 2022. So you don't have to realign or recalibrate the way you have been preparing for NEET PG so far. Please go ahead with whatever you have been doing so far. Okay, but yes, I know, Dr. P, yes, it was very tough, plus but you don't have to worry about this as far as NEET PG is concerned. That will be a totally different scenario. Okay, so let's try discussing all the questions now. And before we start discussing all the questions, let me tell you few facts about Unacademy plans. Let me start with the good news. There is a learning festival that is happening on Unacademy platform. So for the next 15 days, Unacademy Plus subscription would cost you only $299, and this is valid between May 9th and May 16th. So try to utilize this offer. This is like never before. So if you're asking me what does Plus subscription entitle me with, so if you have a Plus subscription, you will be able to access all the structured courses which are planned on the platform. And currently we have an FMG 2022 High Yield Revision MCQ batch which is starting on May 11th. And in this batch, all 19 subjects will be discussed by top educators. And we have also a batch on UPSC CMS revision and PYQs. And in this, subjects covered would be OBG, Pediatrics, Surgery, Medicine and PSM. And this is about the UPSC CMS plan. And Plus subscription will also include MBBS Prof 1 course. And MBBS Prof 1 course is programmed for the first year MBBS students who have just joined MBBS. And in this course, anatomy, physiology, biochemistry, all three subjects will be discussed in detail. And it will also be aligned clinically. It will be aligned and integrated clinically. Guidance will also be given regarding preparation for practicals and viva. So this would be the right path that you can guide your juniors to if they are looking for a way to get ready for the first MBBS university exam. And apart from Plus subscription, we also have Light subscription. Light subscription will help you or will enable you to utilize all the practice tests which are happening on the platform. Currently, we have 450 plus practice tests and 90 plus grant tests. And the Plus subscription and Light subscription will enable you to use the previous year question bank section. And this section can be accessed through self-study practice section. And this includes the last 4-year question papers of NEET PG, INI-CET and FMG. So this will help you in quick revision. When I say questions, it's not just questions, explanation will also be provided. Okay. And this is about the All India mock test. So don't miss this test. This is on May 14th. It is at 9 a.m. And this will help you benchmark your performance because this comes along with All India rank prediction. And to enroll yourself for this All India mock test, if you need a code, you can use my code, which is Shanmugapriya. And this is about the NEET PG Practice League that is happening, that has started on May 7th. It will end on May 15th. And every day at 10 p.m., all subjects will be discussed by top educators. And this is the current NEET PG subscription plan. So the minimum that you can opt for plus subscription would be 2 months. That would cost you 7,650. If it's iconic subscription, minimum 6 months, that would cost you 36,000. Light subscription, minimum duration is 2 months, that would cost you 2,160. And all these costs are inclusive of 10% discount. And to get this 10% discount, you can use my code C Shanmugapriya. So that's all about Unacademy plans. Let's now start discussing MCQs. So do you all remember this question? Sanger used dideoxy-NTP for sequencing instead of deoxynucleotide triphosphates because. So what do you think is the right answer? How many of you think you know the right answer? Dideoxy-NTPs exhibit higher affinity for DNA polymerases, lower affinity for DNA polymerases. Insertion of dideoxynucleotide triphosphate results in, you remember this? Good, Duri Ravi Shankar. Can you tell me what you think is the right answer? Yes. Answer is choice C. Insertion of dideoxynucleotide triphosphate results in prevention of elongation of chain by DNA polymerases. Yes. Answer is C. It has got nothing to do with affinity for DNA polymerases. So let me tell you about Sanger sequencing in a nutshell. Yeah, all of you listen to this. Yes, I had already taught. Yes. This has been, this we discussed in high yield MCQ session, right? I think I told you this twice. So Sanger sequencing is based on dideoxynucleotide triphosphate method, otherwise called the termination method. And the principle that is used here is during chain elongation, by mistake, if dideoxynucleotide gets inserted, there can be no further chain elongation. Do you all understand this? Yes, in those 25-hour session. So by mistake, if dideoxynucleotide gets inserted, there can be no further chain elongation. Or in other words, the principle that is used is insertion of dideoxynucleotide triphosphate results in prevention of elongation by DNA polymerases. So why does this happen? To understand this, you should know basics of deoxynucleotide structure and basics of dideoxynucleotide structure. So what is the structure of deoxynucleotide? You don't have to know that in detail. Just have a look at this image. Deoxynucleotide means the second carbon has lost its oxygen. Do you all see that? The second carbon has lost its oxygen. So the second carbon does not have OH. Instead, it has got H. But what is intact? The hydroxyl group at the third position is intact. So what will it enable? This will enable the 3' hydroxyl group to form 3' 5' phosphodiester linkage formation with the next nucleotide. All of you tell me, what is the linkage that links individual nucleotides in a polynucleotide chain? It is 3' 5' phosphodiester linkage. Formation of that linkage is possible only when there is a hydroxyl group at the third position. And that is possible if it is a deoxynucleotide that gets inserted during chain elongation. Whereas what happens if it is dideoxynucleotide? Dideoxynucleotide means both the second and the third carbon atoms have lost their oxygen. In that case, can you form 3' 5' phosphodiester linkages? All of you, can you answer this? In this case, can you form 3' 5' phosphodiester linkages? No. So whenever dideoxynucleotides get inserted by mistake, no further chain elongation can happen. Now, how can this simple mechanism, it's a very simple fact, right? So how do you think such a simple fact help in, how can it help in sequencing? For understanding this, use this image. Yeah. So in this image, the lavender color oligonucleotide is what we are trying to sequence. And whenever you are trying to sequence any oligonucleotide, step number 1. What is step number 1? You subject this oligonucleotide to polymerase chain reaction and you get multiple copies. And after you get multiple copies, you add equal number of copies to 4 test tubes. Do you see that they have shown you 4 test tubes? And in all the 4 test tubes, you add the normal requirements of PCR. Can anybody tell me what are the normal requirements of PCR? Tilly Heavens, Duri Ravi Shankar, anyone? What are the normal requirements of PCR? PCR will always need a DNA polymerase enzyme. And not only DNA polymerase, you know a polymerase will need a primer. So you have to add primers. You will have to add all 4 deoxynucleotide triphosphates, right? After primers, deoxynucleotide triphosphates. You need magnesium or manganese to act as a catalyst. You need a buffer. So all these requirements of usual PCR, very good all of you, I'm happy that you all remember. So you add normal requirements of PCR to all 4 tubes. And these 4 tubes differ with respect to which dideoxynucleotide is getting added. For example, in the first tube, if you add dideoxy-TTP, in the second tube you add dideoxy-ATP, third tube you add dideoxy-GTP and dideoxy-CTP. So how do these 4 tubes differ? These 4 tubes differ only with respect to which dideoxynucleotide is getting added. Okay, now let's understand what happens in the green tube. I am just taking it as an example. So let's understand what happens in the green tube. What should be the polarity of the template strand? Polarity of the template strand is 3', 5'. So start from the 3' end. What do you have here? You have A. And across A, what will a DNA polymerase attach? It will attach T. And across G, it should attach C. Right? Across G, it should attach C. When it has to attach C, there are 2 probabilities. This tube has got both deoxy-CTP and dideoxy-CTP. So it's just equal probability. Suppose you have added 100 copies of these oligonucleotide. Out of these 100 copies, to 50 copies it attaches the usual deoxy-CTP. When it attaches the usual deoxy-CTP, elongation continues. But to the remaining 50 copies, it is going to attach dideoxy-CTP. If it attaches dideoxy-CTP, elongation stops with that. And if elongation stops with that, that is going to produce an oligonucleotide of length what? It is going to produce an oligonucleotide of length 2 nucleotides. Can you see that? It produces an oligonucleotide of length 2. Now to the remaining 50 copies, when it attaches deoxy-CTP, elongation continues. Okay, elongation continues until against the 8th nucleotide. 8th nucleotide is again G. Against this G, it attaches deoxy or dideoxy. When it attaches dideoxy, it stops with that. So that will provide an oligonucleotide of length 8 nucleotides. So this funnel or this tube will have 2 oligonucleotides, one of length 2 bases, the other of length 8 bases. And that you will be able to find out when you run it through electrophoresis. So when you run it through electrophoresis, you know that if it moves till this, the length is 2 bases. And if the length is 2 bases, it means as the second nucleotide. If the length is 2 bases, it means as a second nucleotide, it has attached dideoxy-CTP. Why will it attach dideoxy-CTP as the second nucleotide? Yeah, why will it attach dideoxy-CTP as a second nucleotide? Because the template has got G. So this tells you that the second nucleotide in the template is G. Okay, now look at this. If it moves till this, you know its length is 8. And if its length is 8, it means as the 8th nucleotide, it has attached dideoxy-CTP. Why will it attach dideoxy-CTP as the 8th nucleotide? Because in the template, it is G. Do you understand this? It means it is in the template, it is G. So just by looking at one tube, just by looking at one tube, you now know the second nucleotide and the 8th nucleotide in the template is G. Do you all understand this? Just by looking at this, you know that the second and the 8th nucleotides are G. So have you sequenced Gs? So that way, if you integrate the results of all the 4 tubes, you will know which nucleotide should be placed where. And this is how Sanger sequenced the oligonucleotide for the first time. Do you all understand this? So what are the facts that you need to know about Sanger sequencing? Sanger sequencing is nothing but dideoxynucleotide triphosphate method. It is otherwise called as termination method. Yeah, it is otherwise called as dideoxynucleotide triphosphate method or termination method. What is the principle that is used? The principle that is used is, by mistake, if dideoxynucleotide gets inserted, no further elongation can happen, or DNA polymerase will not be able to continue the elongation process. So answer for this question is choice C. Okay, so this was discussed in our high yield MCQ session when I did biochem in 25 hours. Okay, so it must have been easier for you all. Now this is the next question, which I thought is very interesting. I was like a bit happy to see such questions. All these are conceptual questions. Okay, so whenever I discuss in the class, when I say please switch over to factual to conceptual learning, do you understand that? Do you understand the significance of it? And this question is not new to AIIMS. This was previously asked long back. Yeah, this was actually a PGA question. To begin with, it was a PGA question, and then AIIMS had this question. Now INI-CET has asked the same question. Yes. So affinity maturation of antibodies is because of what? It is because of somatic. No, it is not because of differential mRNA processing, Duri. It is because of somatic hypermutation. Yeah, all of you listen to this carefully. You will understand the interesting aspect of how you produce antibodies. Okay, so no point just memorizing answers anymore. Try to learn it conceptually. So differential mRNA processing will help in isotype switching. Have you all heard of isotype switching? What do you mean by isotype switching? You initially producing IgM for primary immune response, then switching over from IgM to IgG for secondary immune response, and then producing IgA if it's a surface immunity which you want. That is called as isotype switching. And isotype switching is done by differential mRNA processing, whereas affinity maturation is by somatic hypermutation. So I will take another 5 minutes for me to tell you all about how you produce antibodies. I want you all to listen to this carefully. Okay, so before we start discussing about that, I want to quickly recall. I know you all know this. Structure of antibody is, every antibody will have 2 light chains and 2 heavy chains. And both light chain and heavy chain, they have variable regions, right? They have variable regions and constant regions. Now, what does variable region determine? Variable region determines antigen binding sites. Yeah, that determines antigen binding sites. Whereas constant region will determine the type of light chain or the type of antibody. For example, the constant region will determine whether the light chain is of kappa type or lambda type. And the constant region of heavy chain will determine whether the antibody is going to be of IgM, G, A or D or E. That is going to determine which type of antibody is it. If the constant region is of mu type, you produce IgM, gamma type it is IgG, alpha type is IgA, delta type is IgD, epsilon type is IgE. Do you all remember that? So the constant region determines the class of antibody. So this is an introduction to structure of immunoglobulin. And if you look at the gene which represents the heavy chain, yeah, the gene which represents the heavy chain, it is represented on the long arm of chromosome. And on the long arm of chromosome 6, if you see how it is arranged, starting from the centromeric region to the telomeric ends, you have various parts of heavy chain represented. Do you all know that heavy chain has got V, D, J and C regions? All of you, V, D, J and C regions, variable region, diverse region, joining region and constant region. And all these regions will be represented in the long arm of chromosome sequentially. Close to the centromere is V locus, then D locus, J locus and constant locus. And what is interesting is V locus alone has got 100 alleles. Yeah, V locus alone has got, yes, VDJ rearrangement happens. So that's what I'm going to tell you now. So V region alone, V locus alone has got 100 alleles. Why do you want so many alleles? It is because depending upon the antigen to which the B cell is going to react. depending upon to which antigen the B cell is going to react, the V region should be altered. So V region alone has got how many alleles? There are 100 alleles. And D, J and C loci each will have 5 alleles. Yeah, each will have 5 alleles. What are the 5 alleles of the constant locus? Constant locus, just now I told you, what are the alleles that you can expect? Mu. Close to the centromere is mu allele, and then it is gamma allele, alpha, delta and epsilon. Do you understand this? This is how it is arranged. Close to centromere it is mu, then gamma, alpha, delta and epsilon. So can you tell me how is this immunoglobulin heavy chain gene different from other genes? In all the other genes, the allele is being already chosen. Do you all understand this? In all the other genes, the allele is already chosen by the time the person is born. But immunoglobulin heavy chain gene alone, you don't choose the allele at the time of birth. So after birth, depending upon what allele is chosen, you get various clones of B cells. Do you understand this? Depending upon which allele is chosen, you will get various clones of B cells. For example, if a B cell decides to react to an antigen, depending upon the antigen to which the B cell is going to react, that particular V allele will be chosen. For example, this B cell has decided to react to an antigen for which V3 allele is what we want. So that particular V3 allele alone will be chosen. That is combined with D allele. Let's have it as D3 allele, J3 allele. I'm just giving you an example as 3 because 3 is my favorite number. I mean, no other significance. I'm just giving you an example. So V3, D3, J3. So all these have been chosen. And after these are chosen, the remaining alleles are unwanted. Yes or no? The remaining alleles are unwanted. So those alleles get deleted. And these V3, D3 and J3 will be rearranged. And after rearrangement, they form a single transcription unit. And only after they form a single transcription unit, this particular B cell will be capable of producing an immunoglobulin. And which immunoglobulin will it produce first? Which allele is present first? Mu allele is present. So if you transcribe mu allele, that is going to be called the surface IgM. If mu allele is transcribed, that will be called the surface IgM. And the moment this B cell starts expressing surface IgM, yeah, it is going to be called as a B cell. Until then, you will call it as a stem cell. Because what is the requirement for a stem cell to be called as a B cell? It should have a receptor for antigen. What is the receptor for antigen? It is surface IgM. And only when a stem cell becomes capable of expressing surface IgM, you will call it as a B cell. And what is essential for a cell to be able to express surface IgM? That is called as VDJ rearrangements. Have you understood this? So what is the first step that happens in immunoglobulin? What is the first step that happens in B cell development? In B cell development, the first step that happens is VDJ rearrangements. Have you understood this? Yeah, it is VDJ rearrangement. Following VDJ rearrangement, the cell becomes capable of expressing which antibody? It starts expressing surface IgM. The moment it starts expressing surface IgM, that is the antigen receptor. So now you will call it as a B cell. Okay. And after it starts becoming a B cell, depending upon which type of immune response you want, you should be capable of switching between various antibody production. If it's a primary immune response you want, then the B cell should be capable of producing secretory IgM. Can anybody tell me what is the difference between surface IgM and secretory IgM? Any one of you? What is the difference between surface IgM and secretory IgM? Surface IgM will have an adsorption site. Isn't that logical? Yeah, surface IgM should have an adsorption site. Once the adsorption site is missing, it becomes a secretory IgM. Okay, so it is adsorption site that differentiates surface IgM from secretory IgM. So how will you be able to produce secretory IgM? So as always, B3, D3, J3 allele have been chosen. Following VDJ rearrangement, it has become a transcription unit. So all these will be transcribed, and the mu allele will also be transcribed. But the poly A tail gets added well ahead. Do you all understand this? The poly A tail gets added well ahead. So what will not be allowed to be synthesized? What will not be synthesized? The adsorption site will not be synthesized. Once the adsorption site is missing, what will you call it as? Secretory IgM. So alternative polyadenylation site. What do you call it as? Alternative polyadenylation site. Preponing poly A tail addition will help in the conversion of surface IgM to secretory IgM. Clear? And then if it's a secondary immune response that you want, then you have to produce IgG. How will you be able to produce IgG? B3 allele, J3, D3 allele, J3 allele will be transcribed. Mu allele and gamma allele will also be transcribed. After transcription, poly A tail gets added. And mu sequences will be treated as introns and they will be spliced out. Do you understand this? Mu sequences will be treated as introns and that will be spliced out. Once you remove mu sequences, what is the type of antibody it is? It is IgG. So what will you call it as? This is called as alternating splicing sites. What will you call it as? It's called as alternating splicing sites. That is how we are going to produce the various classes of antibodies. Similarly, if you want surface immunity, mu allele will be transcribed, gamma will be transcribed, alpha will be transcribed, and then poly A tail gets added. And mu and gamma will be spliced out. And what will it produce? IgA will be produced. This is again alternating splicing sites. So can you call these two? Yeah, alternating polyadenylation site and alternating splicing sites. Can we call these two as differential mRNA processing? Together, can you call it as differential mRNA processing? So this differential mRNA processing helps in what? It helps in isotype switching. What do you mean by isotype switching? The variable site is maintained. Yeah, the variable site is maintained. Only the constant site is getting altered. That is called as isotype switching. That is possible by differential mRNA processing. Okay, so all these happen even before the B cell is getting exposed to an antigen. Now for the first time the B cell is getting exposed to an antigen. The antigen comes and binds to surface IgM. The moment a particular antigen comes and binds to surface IgM, thereafter that clone of B cell will be committed only for that antigen. Okay, and when I say you choose V3 allele, all that you would have chosen is, you would have chosen the class of antigen to which the B cell is going to react. And that class of antigen will have thousands of antigens. Now it has gotten attached to one specific antigen. Thereafter the B cell should be specific for only that antigen. So you have to improve the specificity. What do you mean by improving the specificity? That is called as affinity maturation. How will you improve the specificity? By introducing hypervariable regions. Already there are variable regions. In the variable region, you are going to introduce hypervariable regions. So how will you introduce hypervariable regions? In the chosen B3 allele, in the chosen B3 allele, you induce mutations. You induce mutations in such a way that this V region becomes more specific for that particular antigen. So you are inducing mutation in a somatic cell. You are inducing mutation in a somatic cell. That is called as somatic hypermutation. Do you all understand this? That is called as somatic hypermutation. So this somatic hypermutation helps in affinity maturation of B cells, because that helps in you introducing hypervariable regions in the variable region. So now do you understand the answer? Somatic hypermutation causes affinity maturation of B cells. So can we summarize all that I've told you so far? Yeah, can you help me summarize? The summary is, the first point is VDJ rearrangement. The first step in B cell development is BDJ rearrangements help in the conversion of stem cell to a B cell. After stem cell becomes B cell, isotype switching, the ability to produce various classes of antibodies by a clone of B cell, that is done by alternative mRNA processing. So alternative mRNA processing helps in isotype switching. Okay, and then specific affinity for an antigen, that is called as affinity maturation, and affinity maturation of antibody is done by somatic hypermutation. So will you be able to answer such questions in future? Yeah. So that is the answer for this question. Affinity maturation of antibodies because of what? It is because of somatic hypermutation. Okay. So this is another question which was asked. Which of the following is false about Human Genome Project? What do you think is the right answer? It took more than 2 decades to complete the genome. Is that true? Yeah, because the latest draft of Human Genome Project, including Y chromosomes, yeah, came up only in the year 2022. Is it not interesting? It started in the year 1990. Immunity is always a bit tough. Yeah, it is not a bit tough, it's conceptual. Yeah, only when you try to skip the concept and try to memorize fact, it looks as if it's tough. So I strongly recommend that please don't do factual learning alone. I'm not saying don't remember facts. I'm saying remember fact is step number 1, but don't stop with step number 1. Progress to conceptual learning. Try to understand why. Yeah, why do we say somatic hypermutation? What do we mean by affinity maturation? What do we mean by alternative mRNA splicing? So ask yourself why behind every fact that is being told to you. Understand everything conceptually. Only then you will be able to answer such questions. Okay? Only then everything will make sense. Otherwise, it's like just memorizing and forgetting. Okay, so does it make more than 2 decades to complete the genome? Yes, it took more than 2 decades because the latest draft came in January 2022, including the sequences of Y chromosome. International collaboration was utilized. Yes, international collaboration was utilized because it was an international science project. Though it was started in US, then UK, and all countries joined, and then they finally did this Human Genome Project. So A and B are true. Number of genes identified is 20,500. Do you think that is true? Number of genes identified is 22,300. Yeah, it is 22,300. So this is the right answer. Which is false about Human Genome Project? Number of genes identified is not 20,500, it's 22,300. Next generation sequencing was used? Yes, only because we started using next generation sequencing with long reads. Long read in the sense even longer polynucleotides can be sequenced by next generation sequences, which cannot be done by Sanger sequences. So when I say next generation, it means it is not Sanger sequencing, the next level. And that was used in Human Genome Project. Okay. So let me tell you quickly few facts about Human Genome Project, because they are not going to exactly repeat the same question. Okay, so Human Genome Project was started in the year 1990, and when it was started, it was headed by Francis Collins. Yeah, it was headed by Dr. Francis Collins, and this was funded by National Institute of Health US. And this was in 1990, and in 1998 there was somebody who was challenging this. Yeah, it was a private company, Celery Genomics. So Celery Genomics, which was headed by Dr. Craig Venter, in 1998 challenged this project, and they said they will be able to sequence it faster. So that competition made everything quicker. And at one point, both of them collaborated. And because they collaborated, they could come up with the first draft well ahead of the target. The target was only 2010. Yeah, the target was only 2010. They thought 2 decades it will take. But they came up with the first draft in 2003. Yeah, they came up with the first draft in 2003 when they sequenced 92% of the genome. And only this they could do. They were not able to sequence highly repetitive sequences. because if it's highly repetitive sequences, Sanger sequencing will be very difficult to use. So they had to stop with 92% of genome. Then we started getting next generation sequencers, so we started doing more and more research on it. And so in 2021, the title complete genome was assigned. Complete genome, wherein less than 0.3% could not be sequenced. Even that was not exactly complete, though we called it as complete. Less than 0.3% were yet to be sequenced. And only in 2022 January, even the Y chromosome was sequenced. Okay, so how long has it taken? 1990 to 2022, it is 32 years. So it has taken more than 2 decades. Do you understand that? Yeah, it has taken more than 2 decades for sure. And then if you ask me whose genome have we sequenced, did we choose one individual and we sequenced? No, we did not do that. We chose around 270 individuals. They chose around 270 individuals covering all ethnic groups. So Asian, Caucasian, African, European, so 270 individuals were chosen. And then they excluded based on the quality of the DNA that was extracted and based on the health status of the individual, they excluded. And finally they had, it's mostly correct, thank you Mednomic. So finally they had like 20 men and 20 women. Out of these 20 men DNA and 20 women DNA, randomly 2 men DNA and 2 women DNA were chosen, and that was sequenced. Okay, and initially when it was done, Celery Genomics was using only shotgun sequencing method. And in shotgun is shooting, shooting the DNA into multiple fragments using restriction endonucleases, getting shorter fragments and sequencing those shorter fragments, and based on overlapping sequences joining them all together. That is called as shotgun sequencing method that was first used. Only CKA nahi hoga. Okay. Yeah, shotgun sequencing was used, and then next generation sequences came up with long reads. So that is why we have been able to sequence the complete genome. Okay. Now what are the facts or numericals that are related to human genomic sequences? Tell me, what is the total number of base pairs per human haploid genome? Total number of base pairs per human haploid genome is 3.3 into 10 to the power of 9. Yeah, it is 3.3 into 10 to the power of 9 base pairs per human haploid genome. Total number of genes identified is, as I told you, it is 22,300. Yeah, it is 22,300. Yes. An average size of a gene. So if you divide 3.3 into 10 to the power of 9 by 22,300, average size of a gene should have been 27,000 base pair. But average size of a functional mRNA is only 2,000 nucleotides. Yeah, it is only one option was 30k genes in Human Genome Project. No, it's definitely not 30k. 30k was long back. The number has reduced. It is no more 30k. Okay. So average size of a functional mRNA is 2000 nucleotides, which means what is remaining? Remaining are non-coding sequences. Do you understand this? Remaining are non-coding sequences. And if the question is which is the largest gene, can anybody give me the answer for that question? Which is the largest gene? It is RB-FOX1 homolog 1. Yeah, it is RB-FOX1 homolog 1, which is the largest gene. It is RNA binding protein FOX1 homolog 1. I know Mednomic, it was not too tough. I would say every question included multiple concepts. For you to answer every question, you were expected to know multiple concepts. And when we are not that strong in concept, then it becomes difficult. Okay. Titin is not the largest gene, Mednomic. Dystrophin is not the largest gene. Largest gene is RNA binding protein FOX1 and homolog 1. Titin is the largest protein. Do you understand this? Titin is the largest protein. Gene is not the largest gene. But if the question is which gene has got the maximum number of exons, which gene has got the maximum number of exons, then that is titin. Which gene has got the largest exon? That is again titin. Do you understand the answer? Yes, largest number of exons, the largest exon would be titin. And which gene does not have introns? The gene without intron is histone and cytochrome B. The gene without intron is histone and cytochrome B. So these are the facts that can be asked you in NEET PG. Okay. So what is the answer? Which of the following is false about Human Genome Project? It is not the, this is not the right number of genes. Yeah, I will do that mednomic. I will do it soon. Okay. And did you all see this question? You are expected to produce a protein of interest. Arrange the following steps in the correct sequence for the same. To produce a particular protein on a large scale, what would be the sequence, correct sequence? So can you make a guess? Can you first lyse a bacterial cell without doing anything to the bacterial cell? No, we are not going to lyse it first. So what do you think would be the first step? The first step would be, do you understand that to produce any protein of interest, it should be recombinant DNA technology? More options, total 7 were there. If you can recollect and tell me, I can include that, I can help you exclude it. Yeah, tell me what were the choices that I have not included in this? cDNA option was something else, not able to recall. Okay. I was told that something was introduced into bacteria. Was it introduction of recombinant vector into bacteria? No. So I will tell you the concept. The ultimate point behind you attending the session is to understand the concept behind all these questions so that you will be able to answer it if something like this is asked in NEET PG, right? Yeah, so can we go ahead and discuss the concept behind this? Yeah, so what is the question all about? The question is all about recombinant DNA technology. And for something introduced, but cDNA or vector not mentioned. Okay. mRNA, was it mRNA? No. If anyone can help me recollect, it will be of great help for you and for me. Okay. So it is all about recombinant DNA technology, and for any recombinant DNA technology, the first step would be to introduce the cDNA or the gene which produces the protein into the bacteria. Yeah, that would be the first step. Let me show this. So for recombinant DNA technology, to produce a particular protein on a large scale, the first step is to get the DNA which codes for the protein. And after you take that DNA, to introduce the DNA into E. coli, you need a vehicle. And what will you call that vehicle as? The vehicle is called as vector. Okay. So to introduce the DNA into the vector, what are you supposed to do? You should cut the vector. Can you see that here? Isolating the gene of interest mentioned. Okay. So that would be step number 1. Isolating the gene of interest would be step number 1. After you isolate the gene of interest, then you form the recombinant vector. So why do you call it as a recombinant vector? Because you have cut the vector and then you have introduced the fragment with gene of interest. So this is called as a recombinant vector because it has combined prokaryotic vector with eukaryotic DNA. Prokaryotic vector with eukaryotic DNA. That's why it's called as recombinant vector. That is why the technique is called as recombinant DNA technology. And then this recombinant vector is introduced into E. coli. And once you introduce it into E. coli takes care of what? E. coli takes care of producing multiple copies of the recombinant vector, and it takes care of expressing the vector. It takes care of expressing the DNA so that what is produced? Protein is produced. So where do you have all the protein? All the proteins are within the E. coli. So what are you supposed to do? To get your proteins, you should mediate lysis of E. coli. Do you understand this? So after you introduce the recombinant vector into bacteria, bacteria produces protein on the large scale, and to get the protein out of E. coli, you should lyse the bacteria. And after lysis, what will you get? You have got all proteins out. Yeah, all the proteins are released out of E. coli. Now there is not only the protein of interest, many other proteins of E. coli will be there in the protein isolate. And from all these proteins, you want only one protein. How will you get exactly what you want? By subjecting it to SDS page. Do you remember? Was there a choice called as SDS page? Yeah, by subjecting it to SDS page. All of you please understand, SDS page separates proteins based on molecular weight. It separates proteins based on molecular weight. So when you're taking a protein of interest, you know what is its molecular weight. Suppose you know that your protein's molecular weight is 105 kilodalton. Suppose you know that the protein's molecular weight is 105 kilodalton. You run it through SDS page along with the protein ladder. And this protein ladder molecular weight is 100 kilodalton. And can you see that the yellow band, yes, SDS was mentioned, right? Yes, that is definitely needed. So this yellow band has not migrated till 100, which means it's heavier than that. So this yellow band has got 105 kilodalton. So that is probably the protein of interest. Do you understand this? That is probably the protein of interest. So after you lyse the bacteria, you get a protein concentrate. That protein concentrate will have all the proteins of E. coli and the protein of interest. Out of all these proteins, to get the specific protein, you run through SDS page along with the protein ladder. That will tell you this is probably the protein. But to know that this is exactly the protein, you subject it to hybridization. Was there hybridization? I heard that hybridization was one of the steps. So you have to subject it to what? Hybridization and blotting. Okay, so what do? You take this gel and you blot it on a nitrocellulose paper. So the separation is now transferred to the nitrocellulose paper. You take this nitrocellulose paper and you subject it to hybridization, wherein you dip the nitrocellulose paper in a solution containing probes. What are the probes? Can you see those red color worm-like structures? What are those red color worm-like structures? They are antibodies. And what are these antibodies? They are antibodies which are labeled with a radioactive substance, and these antibodies are against only the specific protein of interest. So depending upon where the labeled antibody goes and binds, yeah, the fourth band, it will go and probably bind to the fourth band. So if all the radio labeled antibodies go and bind to the fourth band, now you know fourth band has got the protein of interest. Yes, this is called as blotting. Why am I calling it as blotting? Because I am blotting the gel on a nitrocellulose paper. Why am I calling it as hybridization technique? Because I am allowing the paper to be dipped in a solution which has got a radio labeled probe, and that probe hybridizes with the protein of interest. That is why it's called as hybridization technique. So blotting and hybridization will all be steps involved in getting the protein of interest. Do you all understand this? So tell me, what are the steps involved? If you know this outline, however it is twisted, yeah, the words can be different, the phrases can be different, but these steps are not going to be changed. Do you understand this? So tell me, what are the steps involved in what are the steps involved in getting a protein of interest? Can you all type it in the chat box, please? What are the steps involved in extracting the protein of interest? The first step would be getting the DNA, right? Getting the DNA or getting the gene which can code for the protein of interest. After you take the gene, you have to attach to a vector and introduce into bacteria. And after you introduce it into the bacteria, what does the bacteria do? Bacteria will clone it, will express it. So within the bacteria, you have all the proteins in excess. What should you do? You should lyse the bacteria so that the protein comes out. When the protein comes out, it's a complex protein solution, but you want only one protein. How will you get exactly what you want? By subjecting it to SDS page. SDS page separates proteins based on what? Molecular weight. So molecular weight separation will tell you probably this is the protein of interest. And then to make it very specific, yes, this is the protein of interest, you subject it to hybridization technique. And this is called as Western blot. Why is it called as Western blot? Because using this, you are trying to identify a protein of interest. So this is called as Western blot. So these are the steps that are involved in identifying or extracting a protein of interest. So can you tell me, out of these steps, can you tell me what is the sequence? Yeah, out of these steps, the first step would be, as you said, extracting or isolating the DNA. First step would be isolating the DNA which codes for the protein of interest. After you isolate it, you introduce it into bacteria. After you introduce into the bacteria, you lyse the bacterial cell, from where you get protein, and that protein is subjected to SDS page, and this SDS page is followed by hybridization and elution. Yeah, that is subjected to hybridization and elution. Is that clear, all of you? So these are the steps that we usually do to get a protein of interest in the laboratory. Now the remaining questions are quite easier. Deamination of methylated cytidine residues results in incorporation of which of the following into DNA? Can you try to tell me this? Deamination of methylated cytidine residues results in incorporation of which of the following? Answer is thymidine. Why does it form thymidine? For you to answer this, you will have to know about pyrimidine synthesis. In pyrimidine synthesis, uridine is first formed. What are the pyrimidines you want? You want cytidine and thymidine. But to synthesize cytidine and thymidine, what is first formed? Uridine is first formed. And this uridine on amination gives you cytidine. All of you please memorize it. Uridine on amination gives you cytidine, whereas uridine on methylation gives you thymidine. Okay. Now what will act as a source of this methyl group? Because this is the basis of another question. What will be the source of this methyl group? Source of this methyl group is N5N10 methylene tetrahydrofolate. So you need tetrahydrofolate to convert uridine to thymidine. Is that clear? Okay. So once you have thymidine and cytidine, now the next fact is, yes, one carbon pool. Exactly, it is one carbon pool. Now about epigenetic alterations. What are epigenetic alterations? These are inheritable changes in the DNA, yeah, which are not related to sequence changes. These are inheritable changes in DNA which are not because of sequence changes. And the most common epigenetic alteration that happens in your DNA is methylation of cytidine residues. This has been asked so many times. It is methylation of cytidine residues of CG islands. What does that mean? In your chromosome, you have 46 chromosomes. And in most of your chromosomes, there are multiple CG islands. What do you mean by CG islands? They are rich in CG sequences. And wherever there are CG islands, these cytidine residues, they prefer to get methylated. And once they get methylated, those regions will not be expressed. Do you understand this? Methylation of cytidine residues of CG islands will not allow that region of chromosome to be expressed. It becomes heterochromatin. This is happening normally. Okay, but these methylated cytidine residues, they have a tendency to undergo deamination. What will happen when they undergo deamination? Have a look at this. Yeah. So this cytidine is undergoing methylation. When this undergoes methylation, can I call it as methylated cytidine? Yeah, I will call it as methylated cytidine. And can you call methylated cytidine as aminated thymidine? Methylated cytidine is nothing but aminated thymidine. So this is undergoing deamination. When this undergoes deamination, what will you get? You will get thymidine. Do you all understand this? So deamination, deamination of methylated cytidine residues will form thymidine. So these regions will get converted to TG. Instead of CG, it becomes TG. Okay, so therefore there won't be any heterochromatin formation. All these start getting expressed more and more, and that can cause cancer. Yeah, that is why they've asked you. They didn't ask you this for no reason. So one of the causes of cancer is deamination. It is deamination of methylated cytidine residues. Deamination of methylated cytidine residues, because of which there is no heterochromatin. Those regions get overexpressed. Proto-oncogenes get overexpressed. That can cause cancer. Okay, so what is answer? That results in thymidine formation. Now can you answer this question? Misincorporation of uracil into DNA is because of deficiency of what? instead of thymidine, yeah, instead of thymidine, if uracil gets incorporated, then what is the cause? I told you, for you to convert uracil to thymine, you need tetrahydrofolate. So instead of thymidine, when will uracil get incorporated? Whenever there is folate deficiency. So answer for this question is choice C. It is folate deficiency. So I want you all to remember that tetrahydrofolate uses are, it's a part of one carbon pool wherein it acts as a source of one carbon derivatives like N5, N10 methylene tetrahydrofolate, which helps in the conversion of uridine to thymidine. So whenever there is folate deficiency, uridine accumulates and that gets misincorporated into DNA. B12 was also mentioned in options. That can also cause the same, because B12 deficiency, yeah, because B12 deficiency can cause methylfolate trap. You know that, right? B12 deficiency can cause methylfolate trap, and that folate deficiency can also result in the same disorder. Okay, but given a choice, what will you choose? Which has got a direct effect? For B12 deficiency to cause folate deficiency, there is one step. And then this folate deficiency causes misincorporation of uracil. So what will you choose? Even when both are given, I would go for folate deficiency. Do you all understand this? I would go for folate deficiency because it takes some time for B12 deficiency to present with methylfolate trap. Clear? So did you all see this question? DNA protein interaction can be studied using. Yeah, DNA protein interaction can be studied using. The answer is DNA footprinting. Yeah, answer is DNA footprinting. So first tell me, what is DNA fingerprinting? DNA fingerprinting is nothing but person identification. It is nothing but person identification by utilizing uniqueness of DNA sequences. Do you understand this? DNA fingerprinting is like dactylography. What is dactylography? Person identification by utilizing uniqueness of fingerprints. Now what is DNA fingerprinting? It is also person identification by utilizing uniqueness of DNA sequences. And this DNA fingerprinting was first identified by Jeffrey. When Jeffrey identified it, he used RFLP. So tell me, DNA fingerprinting uses what? It uses restriction fragment length polymorphism, or we use microsatellite repeats and mini satellite repeats for person identification. The other name for mini satellite repeat is variable number of tandem repeat analysis. So suppose in NEET PG question they ask you, person identification is done by, then what will be your answer? Your answer will be restriction fragment length polymorphism or microsatellite or mini satellite analysis or VNTR analysis. Clear? So that is not for DNA protein interaction. Similarly, this is not northern blotting as well, because what is northern blotting used for? It is just used for identifying unknown RNA sequences. Southern blotting is used for identifying unknown DNA. Northern blotting is used for identifying unknown RNA. Western blotting is used for identifying unknown protein. So all these uniquely identify DNA or RNA or protein. They are not going to identify DNA protein interaction. Okay, so it is not northern blot as well. And this is not ELISA because DNA footprinting is also provided in the choice. Okay, because otherwise ELISA, what is ELISA? It is a immunoassay. What are immunoassays? Any assay which is based on antigen antibody reaction. Now what is ELISA or enzyme linked immunosorbent assay? In this case we use an enzyme labeled antibody or an enzyme labeled antigen to find out a particular protein. Do you understand this? In this, what do we use? We use enzyme labeled antigen or enzyme labeled antibody to find out a particular protein. And this can be used to find out protein, but it cannot be clearly used for identifying DNA protein interaction. But this field is progressing. A study on DNA protein interaction is progressing. And now they are coming up with ELISA assays to quantitatively detect and tell you how much of DNA protein interaction can happen. But DNA footprinting is the ideal method. Yeah, DNA footprinting is the ideal method that is used to study DNA protein interaction. Okay, so what are the methods that can be used to study DNA protein interaction? The most commonly used method is based on the fact that after DNA binds to a protein, electrophoretic migration will be altered. After a DNA binds to a protein, the DNA's electrophoretic mobility will be altered. So that method is called as electrophoretic mobility shift assay. Yeah, what do you call it as? I didn't know an entity called as DNA footprinting existed. Yeah, so this is quite a complex question, and undergraduate is not expected to know this, but they have asked you, so this means you have to start reading about it. Okay, so DNA protein interaction. The most commonly used method is electrophoretic mobility shift assay. It is based on the fact that after DNA binds to protein, its electrophoretic mobility will get altered. So all that this electrophoretic mobility shift assay can tell you is, okay, is there any protein interaction or no protein interaction? It will tell you yes or no. There is an interaction, no, there is no interaction. Okay, but it will not tell you details. What are the details that you want? You want to know to which sequence of the DNA does this protein go and bind. You want to know to which sequence of the DNA the protein goes and binds, and for that we use DNA footprinting. Do you understand this? So what is the use of DNA footprinting? It helps you in identifying the sequences to which the protein goes and binds. Now what is SELEX? SELEX is the latest one. So SELEX is, what does it help in? SELEX helps in identifying oligonucleotides. Out of the various oligonucleotides that you have synthesized, to which oligonucleotide does this protein have a specific affinity? Yeah, what is SELEX? SELEX is used to, you have mentioned it in one of your classes once. Thank you. Yes, Gayatri. I have almost mentioned all these facts in all my sessions, but I do understand that most of you would not have attended all my sessions. So you think it is difficult, but actually all these have been already discussed. Okay, Harper, no, it is not mentioned in Harper. Okay, so but all these techniques we have already discussed in all our sessions. I have discussed everything conceptually. So what is SELEX? SELEX is systematic evolution of ligands by exponential enrichment. So you don't have to know exactly what it means, yeah, but for you to answer such questions, you have to know the moment the question is DNA protein interaction, you should start saying the most commonly used is electrophoretic mobility shift assay. Most commonly is electrophoretic mobility shift assay, which just tells you whether there is a shift or no shift, whether there is an interaction or no interaction. And then to get more details, to which sequence does the protein go and bind, that is done by DNA footprinting. Thinking it does not exist and shows ELISA. Okay. Please don't feel bad about it. Please don't think you are less than others because you made such mistakes. These are not mistakes. These are bound to happen because such complex questions you cannot remember. So that is why my advice to you all is, yeah, before you write your NEET PG exam, the day before NEET PG, please spend some time and understand molecular biology techniques. Because you must have attended many sessions of mine on molecular biology techniques. Whenever I say, you think, okay, I've understood it, okay, I know the answer, but take it from me, you will forget it. Yeah, because these techniques you have never done, you have not handled all these techniques. So all these are by you visualizing what I'm trying to say based on the concepts which I give you. So all these will be volatile. So I want you to revise the day before NEET PG molecular biology techniques. Okay. Sometimes I'm unable to assimilate new topics as I was bonded to previous year questions. Yes. Attending NEET PG, INI-CET, all these are like not, it's mostly inhumane efforts that you'll have to put in. Yeah, inhumane efforts you'll have to put in, and all of you do that. I'm not saying you're not doing it. All of you do that, and after you do that, you are bound to make mistakes. Okay, so you don't have to be harsh on yourself. Nobody would have answered all these questions, so it is not you who have not answered. All of them would have made mistakes in this, so don't feel bad about it. This is not going to affect your rank, but I'm discussing about all these because you should not make mistakes again. For NEET PG, if there is a question based on this, you should be able to answer it. Yeah, that's why I'm telling you all these. Yes, all of you make, all of them make, I make, everybody makes. Though I handle all these techniques frequently, yeah, sometimes when the question is asked, I mean, I thought it was ELISA the moment I saw this, because ELISA is upcoming for quantitatively detecting whether there is an interaction or not. ELISA is upcoming, so I thought ELISA. Then I was told that there was DNA footprinting. If there is DNA footprinting, no other go, you'll have to choose that. Okay, so don't worry. Even known facts got muddled up this time. Don't worry, Tilly. Take this as an opportunity for you to recalibrate your preparation. So the day before exam, have in mind the list of topics that you need to revise the day before exam. Don't randomly read everything. So there are few volatile topics. So on Unacademy, there is a batch which is for frequently forgotten volatile topics batch. So try to attend those plus classes. In those plus classes, I will be discussing all these molecular biology techniques in detail, because I know that's the most volatile of all. Okay, so before the day before exam, again revise it. So that will help you. Please don't be scared, Ibiza. Don't feel negative at all. Be positive. You will be able to do it only when you feel positive. Okay, and again I always say this, it's not just about the mark which you score, it is not just about the rank which you get, it's not just about the seat which you get. It is all about the knowledge which you gain during this period. Yeah, because this is going to be the foundation for you to shine when you get any PG seat. Yeah, so it's not mandatory that everybody gets radio diagnosis, that everybody gets ortho, everybody gets dermatology. All these are sought after courses, right? So I don't have anything against these courses, but not everybody can get that. That we should acknowledge, okay? And that does not mean it's a failure for you all. You can take even a course which nobody wants, yeah, but then you can shine in the course, provided you have a strong foundation. Okay, so take it from me, if you have a strong foundation, any course which you take, you will be the apex. Yeah, you will be the sought after star. Do you want to be the sought after star, or to be one among the million? If you want to be a sought after star, then you have to do a conceptual learning, understand it, cherish the process of learning. Yeah, so how many ever days you have from today before your NEET PG, be happy when you learn a new fact, be happy when you learn a new concept. Yeah, that way it makes you feel positive, and that positive energy is much needed when you are approaching an exam. Okay, so forget about what has happened in INI-CET, don't worry about it at all. Take that as a reason for you to fine tune your knowledge. So now you know what are the topics you are not strong in, right? So you can fine tune those topics for NEET PG. Okay, without you would have been lost. Was at least able to rule out and put my head. I am very happy, Skull, I am happy. Can you please tell important topics to revise the day before exam? I can tell you topics to revise before the day before exam, but I would say only biochemistry and physiology. So it has got person to person variation, Gayatri. So you will have to decide on what are the volatile topics for you. Okay, so those topics you start making a decision from now. So from now, start working out questions, and when you work out questions, you will know these are the topics I'm weak about, I'm weak on or weak at, and these are the topics which I have to revise. So make a note of all those topics, and the day before exam you can do it. Yeah, please don't get demotivated. I know this would happen. Soon after I saw the question, I was upset, not because of the questions, but because I know the impact. I could imagine the impact that would have that this question paper would have on you. That was my major worry. But please don't get upset. You always have multiple chances, multiple choices. Okay, so it's not about just getting into a premier institute. It's not just about getting the sought after course. It is all about the knowledge. Okay, so keep gaining knowledge. Infrequently forgotten, will you discuss physio also? I will try to include few physio topics also, Ibiza. Yeah, I will do that. So this is about DNA protein interaction. So this is the last question. Very simple question. Out of all the questions, I was very happy to see only this one. So in maple syrup urine disease, which of the following need not be limited? Ma'am, please host a session day before the exam for motivation. We need your words and blessings. I will definitely do it, Skull. If not the day before exam, I know you'll all be very anxious. If I say something, you will be like, what is she blabbering? So probably 2 or 3 days before, I will try to have a session. Yeah. So answer is methionine. Yeah, answer is methionine. So branch chain amino acids, maple syrup urine disease is caused by a defect of branch chain keto acid dehydrogenase enzyme. And so branch chain amino acid metabolism is affected. So branch chain amino acid should be limited. Yeah, intake of branch chain amino acid should be limited. So what need not be limited is methionine. Okay. So these are the questions that were given to me by you all, and which I've tried to make replicate what could have been asked in INI-CET, and try to revise all these questions. All the best. Don't get demotivated. Be positive. Yeah. No. I'm sorry. That was by mistake. Okay. So thank you all. Good night.